Sorry about that, guys. It wanted to do a restart at the same time. What's going on? Oh, talking about Drew. Oh, from your COVID symptoms. Hopefully you start feeling back to normal soon. Okay, it's just taking me a second to get logged into Canvas so we can do our attendance. I did a claim just yesterday that was about somebody who had COVID in April, and she had such severe neurological symptoms she couldn't even walk afterwards, which is pretty terrible. COVID it's a beast. Okay. All right, I'm going to share the screen for attendance. I'm going to do attendance now, so please speak up or send a chat when I call your name, okay? Make sure you see me mark you as green. That means you're here. Okay, Kayla.
Here.
Thank you. Reagan.
Here.
Kelsey. Kelsey. Okay. Tana. Stevie.
I'm here.
Okay, awesome. Josh.
Here.
Okay. Joel. I think I saw Joel. I saw you, Joel. I don't hear you, but I saw you. Taylor Lobila. She's not here to appreciate me saying that right this time. Let's see, what was the chat? There was a chat. Oh, come on, why won't it show? What did the chat say? Did I miss somebody? Taylor Lavelle.
It was Joel saying he's here.
Okay, perfect. Taylor Lavelle.
Here.
Okay. Grace. No Grace. Andrew. I know you're here. You were talking. Yep. Amber. I don't know what's going on with this. Hold on, let me see. There we go. Jimmy. I thought I saw that Jimmy logged on earlier. Is he still here?
I'm here. Can you hear me?
Yes, I gotcha. When?
Okay.
Here.
Thank you. Marissa.
Here.
Okay. Jamie. Okay. And Jeanette.
Here.
Morgan. I saw Morgan sign in earlier. I'll give you credit, Morgan. I know you were here.
Her speaker's not working.
Oh, okay. Thank you. Macy. Okay. All right. Let us get back to our lecture. Thank you for your patience. I am so sorry about the technical difficulty there.
Jamie said she was here. She put it in the chat.
Oh, my chat wasn't working for some reason. Let me go back. So Jamie Plague. Thank you. Okay, so you have got credit for being here. All right, now let's go back to digestive system. We're almost done with the digestive system. Oh, there it is. Ava is here. I love your chats. I do. if this would just go to the digestive system like I'm requested. Oh my gosh. Oh, Elijah, you're here. Oh, I didn't go to the bottom. Hold on a second. Oh, I didn't. Thank you. Ayla, are you here? Ava's here. I saw her message. Ayla, are you here? Okay. Oh my goodness. Thank you guys. All right. Let's see if we can actually get back to our lecture. That would be nice. I'm going to close this. Okay, can you see the digestive system? Someone let me know because this has been ridiculous.
Yes.
Yes, okay, yay! All right, we are back in business. Okay. So moving down here to the defecation reflex. That is caused by distension of the rectum, and that stimulates the parasympathetic division. So remember, parasympathetic is rest and digest. Okay, so you're stimulating, so this yellow arrow is representing parasympathetic division, sends that information of the stretching rectum to the defecation reflex center at the cona medullaris of the spinal cord, which then sends back voluntary stimulation to contract and expel the feces. Okay, digestion is the breakdown of food molecules for absorption into the circulation. So there's two types of digestion. I know we're kind of backing up for just a second here. You have mechanical, which breaks down food particles into smaller particles. And then you have chemical, which is breaking down chemical bonds by using digestive enzymes. So both mechanical and chemical start in the mouth. Okay, absorption and transport. When absorption occurs, molecules are moved out of the digestive tract and into the circulatory system, and then is able to be distributed throughout the body. So lipids, this includes triglycerides, phospholipids, steroids, and fat-soluble vitamins. So vitamin D is a fat-soluble vitamin. Bile salts, what happens is bile salts surround the fatty acid and the glycerol to form a micelle. So you have bile salts that surround this fat and form basically kind of like a ball with bile salts surrounding it, forming like the outer shell. Okay, chylomicrons, this is a micelle. This is 90% triglyceride, 5% cholesterol, 4% phospholipid, and 1% protein. So chylomicrons are able to enter into the bloodstream, and then they travel to the adipose tissue for storage. So in the blood, triglycerides are converted back into fatty acids and glycerol, where they're transported to adipose cells as well, which is the bane of our existence, right? Is when when the fat is transported to our adipose cells for storage instead of being broken down, then it's converted back into triglycerides. So it's stored as triglycerides in adipose. Triglycerides are converted into fatty acids and glycerol in the blood, but once they get to adipose, they convert back to triglycerides. So they kind of have they start as triglycerides, they convert to something else while they're in the blood, and then once they get to adipose, they convert back to triglycerides. Okay, we're going to talk about the different types of lipoproteins. Okay, so all lipids carried in the blood are combined with a protein. and that makes them soluble in the plasma. That makes them able to transport in the blood without coagulating. So cholesterol. Cholesterol is 15% ingested through your food, and 85% of the cholesterol in your body is manufactured in the liver and by intestinal mucosa. So most of your cholesterol is manufactured by your body. So you don't actually need cholesterol from your diet. Most of the time, cholesterol from your diet is more than you need and can cause problems later on. And that's why it's really important and smart to watch your cholesterol levels, because just remember that most of the cholesterol in your body is produced in your body, so you don't need it from your diet. Okay, lipids are lower in density than water, and proteins are higher in density. So what this does, if we remember, blood is a lot, the plasma is mostly water. So if we had just lipids floating around, they would congeal and separate from the plasma, and we don't want that to happen. So if we bind them to proteins which are more dense, it basically equals out, so they're going to be able to travel in the bloodstream without separating. Okay, so a chylomicron is 1% protein. This is very low density. VLDL, or very low density lipoproteins, is 8% protein. Okay, so triglycerides removed from the VLDL are stored in the adipose tissue. When that happens, it's converted into low density lipoproteins. Low density lipoproteins, which you may have heard of if you've gotten cholesterol tests before, is 25% protein. And this transports cholesterol to the cells, and cells have to have receptors for these low density lipoproteins. When they have receptors, then the cells will take them in. So the number of low density lipoprotein receptors will decrease once the lipid and cholesterol needs are met. So that would be down regulation. And then you have high density lipoprotein, which is 45% protein. Okay, so we're going to talk about a cholesterol disorder called familial hypercholesterolemia. This is caused by increased levels of low density lipoprotein in the blood. And this can cause the buildup of plaque called atherosclerosis in your blood vessels at a faster rate. And so this can lead to coronary artery disease and heart attacks at very young ages, like your age. A xanthoma is a nodule of cholesterol and lipid that's found under the skin, especially at joints. So the people who have familial hypercholesterolemia will have these nodules at joints and sometimes just under the skin, and you can see them because they're kind of whitish yellowish under the skin. Okay, so what causes this disorder is a mutation in the low density lipoprotein receptor gene. So if the receptor is not able to function, you are not able to take in the low density lipoprotein, so they stay circulating in the blood. And that increases your levels of low density lipoproteins, which increases your chances of coronary artery disease and heart attack. So the severity of this disease depends on whether you have one copy of this mutated gene, which is heterozygous, or if you're homozygous, which means you have 2 copies for that gene. If you are homozygous for the trait, that means you have no receptors and you're not able to take in low density lipoproteins, and the severity of this disease is going to be much higher. The buildup of atherosclerosis and plaques in your blood vessels is going to happen very quickly and at a young age, like teenage, early 20s, very young. Okay. Proteins. Pepsin breaks down proteins into smaller polypeptide chains. So pepsin breaks down protein. Then you have proteolytic enzymes that produce small peptide chains. So it breaks down proteins into dipeptides, tripeptides, and single amino acids. After amino acids have been absorbed into the bloodstream, they are carried to the liver. Okay, so all proteins go to the liver once they've been broken down. All amino acids, are carried to the liver. So where is the absorption occurring? So this slide is really helpful, kind of breaks down where absorption and secretion of substances occur. Okay, water can move in either direction across the small intestine, and it really depends on the osmotic gradient. It depends on the concentration gradient of solutes in the small intestine. If the concentration is high, like after a meal, you're going to have a lot of water movement into the small intestine. But after digestion has occurred and absorption has occurred, water levels are lower, and osmotic levels are lower, so water is able to be reabsorbed. So note, 92% of water is absorbed in the small intestine. So the majority of water absorbed in the small intestine. Ions like sodium, potassium, calcium, magnesium, and phosphate are actively transported through the intestinal wall. Okay, so we are going to just keep plowing along because we are actually, for the first time, right on schedule, if not a teeny bit behind, which is amazing. So let me pull up our next lecture, which is metabolism. Okay. Whoops. Pull up metabolism and get us started on that. Okay, make sure I didn't miss any chats. Okay, we're good. All right. Please work. There we go. So in this chapter, we are going to talk about how food is converted into energy. And that process, there's a lot that's going on. So this is a really strong physiology chapter. So this process is going to involve the respiratory and circulatory and digestive systems. And the respiratory and circulatory system are important for delivering oxygen to the cells that we need for cellular respiration, and then getting rid of carbon dioxide, which is a waste product of cellular respiration. And then the glucose from the food that we eat, once it's been broken down into glucose, is used to help manufacture energy in the form of ATP. Okay. So first, talking about what metabolic activity is. This is when you have organic molecules. So remember, organic is anything that contains carbon. So carbohydrates, proteins, and fats are broken down into their smaller components to get energy. Energy is stored in the form of ATP. So whenever we talk about energy, energy equals ATP. There are 3 cycles that we are going to talk about: glycolysis, Krebs cycle, and the electron transport chain. Metabolism involves all 3 of these. Okay, first talk about some definitions. Metabolism is consisting of catabolism and anabolism. Catabolism, think about catabolism is breaking down. So catabolic. This is where you're converting large molecules into small ones. So you're breaking down substances, and in the process of breaking down substances, you are releasing energy, and that energy is stored as ATP. Anabolism, or anabolic reactions, build up. So these are converting small molecules into larger ones. So this is where you're making new organic compounds. Okay, our nutrient reserves. Triglycerides. We just talked about triglycerides and how they are transported in the bloodstream through lipoproteins. So triglycerides are digested to form glycerol. So triglycerides broken down to glycerol. Glycogen is a storage molecule, and you find this in your muscles and in your liver, and it's a storage carbohydrate, which is an easy energy storage molecule that's broken down into glucose when it's needed. And then proteins, these are the most abundant organic compounds in the body. Just a quick refresher, what happens to your body when you do not get enough protein in your diet? You can chat it, or you can tell us till the class. So what happens when you do not get enough protein in your diet? What happens to your body? Oh, well, muscular atrophy can occur. That's true. That's often from not using your muscles or moving your body much. What the answer I was looking for was, remember when you're protein deficient and it causes swelling in the tissues because you don't have the proteins necessary to regulate water balance. So that's what I was looking for, but muscular atrophy can also occur. Yeah, because if you're not getting enough protein, you're going to start taking it from the muscles. So that can happen as well. Okay. All right, here we go. So carbohydrate catabolism. So remember, catabolic is breaking things down. This is when glucose is broken down and converted into energy in the form of ATP. This is the process of cellular respiration. I expect you to know the equation for cellular respiration. I'm going to write it out. Whoops. I'm going to write it out right now. So it's C6H12O6 is glucose. Oxygen is a molecule, so O2. Carbon dioxide, so that's CO2, plus water, H2O, plus energy in the form of ATP. You need to know this reaction. Okay, so cellular respiration. This chemical reaction happens in 3 steps: glycolysis, the citric acid cycle, and the electron transport chain. So we will talk about each of those separately. So in this process, one glucose molecule is converted to 30-ish ATP, so 30 to 32 molecules to ATP. So I just put 30 as a lowball offer there for ATP. So one glucose molecule can produce a lot of energy. Here is a summary slide of cellular respiration and the 3 steps that occur. This is a good one to study from because it gives you all the major players. We are going to go into more detail than this, but this is a good starting summary. So I like this slide, so please take some time and study this slide. So we are going to get into glycolysis. Glycolysis is when you break down glucose in the cytosol of small molecules that can be used by mitochondria. So the first step, glycolysis, is step number 1, does not require oxygen. And whether it requires oxygen or not is something you need to know. This is anaerobic. Anaerobic means without oxygen. And some of our other steps will require oxygen, and you'll need to know that as well. So in glycolysis, you're breaking down a 6-carbon glucose. So remember, glucose is C6H12O6. So you break that down into 2 molecules of pyruvic acid that are 3 carbons. Oops, I'll stop it. I'm trying to erase and it won't let me. Okay, well, then we're not going to worry about it. Okay. My goodness. The computer problems today. I apologize. It's still doing it. Okay. So glucose is broken into 2 pyruvic acids. Okay. So in this process of glycolysis, here is the reaction. So glucose is broken down. It uses energy. So we're using energy to make energy. So we are breaking down 2 ATP molecules. So there's 1, there's 2, and using energy to convert into pyruvic acid. In addition, we are using electron transport molecules that store protons, essentially. NAD is one of those molecules. NAD is converted into NADH, and so that is storing that hydrogen ion. That will be used later, and we'll talk about how it's used in a bit. So if we look down here at our equation, glucose plus 2 NAD plus 2, well, I don't like this, so plus 2 ATP is broken down into 2 pyruvic acids, 2 NADHs, and 2 ATP. So note that we use 2 ATP to break down glucose, but that ends up making 4 ATP at the end. So with some simple math, so 4 ATP minus 2 ATP gives us a net result of 2 ATP produced. That's where we get that right there. Okay. So looking a little closer at the steps of glycolysis, we have our glucose molecule, and we're only looking at the carbon here. That's all we're looking at is the carbon chain. We're not looking at the hydrogens or the oxygens. We're really interested in what's happening with the carbon. So glucose is taken into a cell, and then during step 1 and step 2 adds phosphate to either side of the glucose molecule, and that forms what's called fructose 1,6-bisphosphate. So bis refers to two phosphates. That becomes important because in the last step of glycolysis, that molecule of fructose 1,6-bisphosphate is split right down the middle. And that produces 2 pyruvic acids, which is a three carbons and a phosphate added to the end. Ultimately. So this intermediate step is glyceraldehyde 3-phosphate, which converts into pyruvic acid. So here is where it continues on to that conversion of pyruvic acid. Please don't get overwhelmed. Some of these intermediate steps, I'm not going to ask you all of the names, but I do want to know that you understand the major steps. So the fact that ATP is used to add phosphate to either side of glucose, what the name of that molecule is before it splits, the fructose 1,6-bisphosphate. And then in this process of glycolysis, this is where your NAD is involved. NAD accepts 2 hydrogen ions, so hydrogen ions, to form NADH. NADH is going to be used later, so that's for another step. These 3 carbon chains go through a series of conversions, which I'm not going to ask you these intermediate steps, but note that through these conversions, you have a total of 4 ATP that are produced. You also have some water that is released. The end result is 2 pyruvates, which is a 3 carbon chain molecule. So you end up with 2 pyruvates, 2 NADHs, plus the net of 2 ATP. Okay. So summary of the glycolysis pathway. You have one glucose molecule, C6H12. I know I keep saying this, but you need to know it. C6H12O6 enters the pathway. This converts into 2 pyruvates plus 2 ATP plus 2 NADH. And so NADH, they shuttle electrons, and that will be really important when we talk about the electron transport chain. So this is the overall summary of what happens, which you definitely need to know for sure. So from this step, the pyruvates that are formed in the cell, are then entering into the mitochondria to continue on with the next step. So this is what a pyruvate actually looks like. Note you have 3 carbons. That's what we're really interested in. It enters into the mitochondria through a transport protein. And let me just double check. Oh, some of my pictures. Oh, there they are. Okay. Just checking something really quick. Okay. So pyruvate enters in through a transport protein because it's too big to diffuse across the membrane itself, enters into the mitochondria. And once it enters, there is a series of steps that occur. And pyruvate is converted into acetyl coenzyme A. So in this process, you lose carbon dioxide, you make another NADH molecule, you use coenzyme A. So if you remember, coenzymes are typically vitamins. A really important reason to have your vitamins every day or get it through your diet, but you want to make sure you're getting your diet, your vitamins. So in this conversion, pyruvate loses carbon dioxide, NADH is formed, and coenzyme A is used to form acetyl coenzyme A. Note down here, coenzyme A is a derivative of a B vitamin, pantothenic acid. Okay, so the next step, once pyruvate has been converted into acetyl coenzyme A, it enters into the Krebs cycle. Krebs cycle is step number 2. So this begins with acetyl-CoA entering into the cycle. The very first thing is you lose your coenzyme A. So you're left with oxaloacetic acid and citric acid. So just so you know, Krebs cycle is also called the citric acid cycle because citric acid is one of the products of this process. So the Krebs cycle happens and it goes through 2 cycles to be complete. So one acetyl coenzyme A has to go through 2 cycles of the Krebs cycle to be complete. And you produce 6 NADH, 2 ATP, and 2 FADH. FADH and NADH are the same. They're those electron shuttlers. The electron shuttles, okay, or they accept hydrogen ions. Okay. So we are going to go through that process now, but note in this cycle, we're just looking at the products or things that are produced in the Krebs cycle, the citric acid cycle. 2 carbon dioxide molecules are made, 3 NADHs, 1, I'm sorry. This is through this is 1 cycle, but the cycle has to go through twice. That's why we'll get the numbers in just a second. And 1 FADH2. So if this cycle has to go through 2 turns, this means you have 6 NADH formed, 2 ATP, and 2 FADH2s. Okay, which means then we'll have, instead of 2 molecules of carbon dioxide, we'll have 4 carbon dioxide. Because we're doing 2 circles, 2 cycles. Okay, so here is a little more detail of what is happening in the citric acid cycle. I knew the dog was going to start barking. I'm sorry. It's the trash. The garbage man is here. Okay. So, pyruvate in the cell enters in, so this is when it enters into the mitochondria. So it goes through a transport protein, enters into the mitochondria, and during that process, pyruvate is converted into acetyl coenzyme A. So in that process, you lose a molecule of carbon dioxide right here, and you use coenzyme A and you make a molecule of NADH. Okay, acetyl coenzyme A I'm curious what, I'm gonna back up for one quick second. Acetyl coenzyme A, so that's a 2 carbon chain. So that 2 carbon chain conforms to make a 6 carbon citrate. And you ask like, why? But that's what happens before the next step. That 6 carbon citrate is converted in this cycle, loses a carbon dioxide. Now you have a 5 carbon chain. You lose another carbon dioxide and a hydrogen ion. So your hydrogen ions go bye-bye. They add to your NADH. Now you have a 4 carbon chain. That 4 carbon chain continues on and goes through the cycle again. You lose another carbon dioxide. Now it's a 3 carbon chain. You lose another carbon dioxide. Now it's a 2 carbon chain. Okay. So you go through 2 turns. So in that process, you are creating how many NADHs?
2.
I'm double checking here. 6. So we have 1, 2, 3. So 3 NADH times 2 cycles. So our 6 NADH. Thank you. Then we have ATP. So how many ATP? Remember the cycle goes twice.
2.
2. Good. Okay, then we have FADH2. How many do we make?
2.
Yes. Good. So in this process, it's not a big energy producer, but it does produce a lot of those electron shuttlers, the NADH and the FADH2. Those are going to be used in the electron transport chain. Really important in the electron transport chain. So in our first 2 steps, glycolysis produces how many ATP? Do you remember? Produces a net of 2. And then in our Krebs citric acid cycle, we produce to ATP. So in our first two steps, we haven't made very much ATP. We've made a total of 4. The big energy producer is the electron transport chain. That is where the majority of ATP is made. It's the very big player in this whole story. Okay, this is a more detailed view of what's happening in the citric acid cycle, also known as the Krebs cycle. So remember, this first step where pyruvate is converted into acetyl-CoA happens in the cell. Everything else is in the mitochondria. Okay. Let's see here. What do I want to point out here? Okay, we'll notice that we're starting with a citrate, which is 6 carbons. We do this cycle times 2. So in our first step, we've lost a carbon. Oh, there it is. We lost our carbon. So now it's a 5 carbon. We lose another carbon. So succinyl-CoA is a 4 carbon. So now you're down to 4 carbons. You go through that cycle again, you're down to 3 carbons. When you lose another carbon dioxide, and then you're down to 2 carbons. So you've been able to break down this 6 carbon citrate molecule down to a 2 carbon molecule, which forms citric acid. I wish they would show the citric acid, but that's okay. So the net totals from the Krebs/citric acid cycle so 2 ATP are produced by glycolysis and 2 ATP are produced by the Krebs cycle. So a total of 10 NADHs is produced by the first 2 steps. So 2 are produced by glycolysis, 2 are produced by the process of converting pyruvate into acetyl-CoA, and then 6 are produced by the Krebs cycle. And then we have our new molecule that we learned about in the Krebs cycle, FADH2, and 2 of those are produced. And so the next steps of the electron transport chain is where we get almost all of our energy, or ATP. Okay, so the electron transport chain. So, excuse me. Sorry, I just got something on my, all right. It's annoying me, got it. All right. So we have a series of proteins embedded in the inner membrane of the mitochondria. So look at our little illustration down here of the mitochondria. You have an outer membrane and an inner membrane. So the electron transport chain is occurring across the inner membrane of the mitochondria. In this process, electrons are passed through these protein cytochrome complexes. So we call them cytochromes. Through this process, the electrons lose energy. When they lose that energy, it's used to pump hydrogen ions from the matrix. So the matrix is on the outside. So matrix, not the movie. From the matrix to the intermembrane space. So if we look, so this is just looking at the electron flow. So this is where we first use our NADH. NADH carries 2 electrons, or 2 protons, because they are traveling together. They flow to the first cytochrome complex and attach to energy electron carriers in the cytochrome complex. Oh, Grace got here. Okay. During this process, as these electrons are being transported, they lose energy. That's why the arrow is traveling down. FADH adds more electrons and is converted to FAD. And during this process, the hydrogen ions that are attached to the electrons go and are pumped outside the membrane. That's where the hydrogen ions go. So if you notice, you're building up a concentration difference of hydrogen ions. You're building up a lot of hydrogen ions on the outside of the inner mitochondrial membrane compared to the inside. Okay, so what is the ultimate outcome of pumping these hydrogen ions out? You're building up a concentration difference, which then run what's called ATP synthase. The hydrogen ions flow through this enzyme and they cause this, it basically causes it to spin like a turbine. It spins, and that spinning produces energy in the form of ATP. And so you have built up this hydrogen concentration difference, and it's able to flow through these transport protein enzyme molecule complexes and causes the conversion of ADP into ATP. So the electron transport chain and the formation of ATP. Oh, okay, we'll probably end on this slide. We're doing really well, guys. We're doing really well. I know this is a little bit of a tough chapter, so I'd spend some extra time studying, okay? So with the electron transport chain, there are a series of steps. So energy is released by electrons. So the electron carriers of FADH2 and NADH use our electron carriers, and that energy is used to pump hydrogen ions outside the membrane. More energy, so initially we're using NADPH, and then we use FADH. So this is NADH. So more energy from FADH is used to pump even more hydrogen ions into that inner membrane space. Those electrons eventually are released into the matrix and form oxygen and, and form water. So those electrons eventually form water, which is one of the end products of cellular respiration. Okay, so looking at our time, we're going to start on Tuesday with hydrogen ion pumps. Let's look really quick at our schedule, make sure we're not missing anything. I like to review that. If I can find it. That is not what I wanted to do. Okay, well, bear with me really quick. If you just came in, please stay so I can get you for attendance. So I am not sure if we have a quiz coming up. That's why I want to double check with you before we sign off for the day. So looking at our schedule. Okay, so we need to go further. We're on the next page. Okay, so there is a quiz on Monday. We will continue talking about metabolism. So your quiz will only be on the digestive system, okay? That's a plenty for you to study for. So your quiz on Monday, I'll have it up by Sunday afternoon, and it will be only on the digestive system, and I'll try to throw in some easy or extra credit questions for you, okay? Any questions before we go?
This might be a dumb question, but
Never
like,
dumb.
what is NADH
It is
and
an electron
SADH?
carrier molecule.
Okay.
So when it carries electrons, the hydrogen ions are attracted to it because of the charge difference. Electrons are negative and hydrogens are positive. So it's really carrying electrons and hydrogen ions. Does that help?
Yes, it
Okay,
does.
good. I know that's kind of a weird molecule.
Was it something that was when I was gone?
No, it's brand new.
Okay, okay.
You're right on time. It's brand new for this chapter. Any other questions? Okay, if that is it, I'll let you guys go. And I hope you have a fantastic weekend. And remember to study for your digestive system. And Grace, are you here?
Yeah, I'm here.
Okay, and just make sure I give you attendance grade for that. Okay, everyone have a great day, and I will get that quiz up on Sunday and send an email out. Adios.